Okay. The last group of receptors, the cell surface receptors that I'm going to talk about is the enzyme-coupled receptors. Remember, the first one was the ligand-gated ion channels, the second was with GPCR, and the third one is the enzyme-coupled receptor. Here it is located also on the cell, and this time itself it's an enzyme. Because in the previous case, in the case of GPCRs, it was not an enzyme, and the first few things, these first few steps, they are not enzymatic. It all goes through the allosteric change. But in this case, it is totally enzymatic. Okay. Now, majority of the enzyme-coupled receptor, or the most interesting enzyme-coupled receptors, are receptor tyrosine kinases. So we are going to see RTKs only. Now, these RTKs are tyrosine kinases, receptor tyrosine kinases, and they always work in dimers, homodimer. Okay, so individually, when they are separate, they are inactive. But when the signal comes, they dimerize, which means they come close, and they become active. Now, in some cases, they activate the targets, but in many cases, they cross-phosphorylate each other, which means each of them is a kinase, but it cannot phosphorylate itself. So what it does, that this one is phosphorylating this, and this one is phosphorylating this. So altogether, it becomes an activated RTK dimer. It's like, there's the story that heaven is somewhere with people with very, very long spoons, and hell is also the same place with people with very, very long spoons. In this case, with this long spoon, this couple is activating itself. So it will be now full of tyrosines. They are phosphorylated. Now, these phosphorylated tyrosines, they also behave as a scaffold. So we have never seen scaffolds, but in molecular biology, there are scaffold proteins. Their purpose is to bring things together, because when things are together, they start interacting. For example, these phosphorylated tyrosines, they are harboring some molecules which has SH2 domain. And previously, we have seen domains. They are mostly expressed from a single exon. They used to be proteins, individual proteins once upon a time, but now they are part of the big protein. So all these proteins, they have a separate SH2 domain, and with their SH2 domain, they connect to these phosphorylated tyrosines. And when they are all together, they are active. And in this case, they are going to relay the signal to the downstream pathway. Now, the RTKs, yes, they do lots of things, but they are specifically important for cell survival, proliferation, or let's say first growth and proliferation, and sometimes differentiation. So if you're going to ever study cancer, RTKs will be one of your main targets. So this is how the signal is turned on from an outer environment signal, extracellular signal, binding and dimerizing the RTK. Remember, in the case of GPCRs, it was just changing the conformation. Here, it's not only changing conformation. There is a post-translational addition of phosphate groups. And remember, in the case of G proteins, just the phosphate group of the GTP is hydrolyzed and the G protein comes back to the original conformation. Here, turning off is not that easy. Sometimes the phosphatase enzymes, for some of them, comes and cleaves these phosphate groups one by one. This is a way of turning off the signal of RTK. But in some cases, it's a bit, I should say, brutal. The signal, the RTK, which is activated here, it is taken inside by endocytosis, which means it's like a receptor-mediated endocytosis. It is taken inside. It meets with the lysosome, and inside this RTK is degraded. So you cannot use it like a G protein several times over and over again. And you not only change it post-translationally, but you totally get rid of the receptor. This also implies how important it is to fight against cancer, because overactivation of this pathway commonly ends up with cancer. Now let's check the downstream pathway. Here with the SH2 domain, this adaptor protein binds to the phosphorylated tyrosine. This adaptor protein activates the GAF. Now I hope you remember GAFs. They are the monomeric GTPase activating proteins. So they're working with the molecular switches. Yeah, here they are. Here is a GAF and here is a GAP. The GAF is removing GDP and induced joining of a GTP. GAP is doing the opposite. It removes the phosphate group, so it becomes inactive. So GAF is activating the monomeric GTPase. And here in this case, we are not talking about the trimeric GTPase like the G protein. Here we are talking about a monomeric GTPase called RAS protein. Now, don't confuse RAS with RAB. RAB is located on the vesicles, and it is important for the tethering protein to get it to the correct endomembrane system target. It can be ER, Golgi, or plasma membrane. This time, it is again a monomeric GTPase, but it is RAS protein. Okay, this RAS is activated with a GAF protein. GAF pushes out the GDP so that a GTP joins, and it becomes an active RAS protein and still attached to the plasma membrane with the small lipid group. Now, next, the RAS protein activates MEP kinase. The names are funny. It's a kinase itself. In the second step, it activates MEP kinase. In the third step, it activates MEP kinase. And in the next one, it induces several things, including mitosis. This is mitogen activated protein kinase. As the name implies, one of the main targets is mitosis. So it does several things, including survival, growth, proliferation. This is why it is very important for the cancer studies, because when this RAS is active, it will most possibly push the cell to divide. Now, currently we know that in many cancer cells, in many cancer situations, the RAS is constitutively active, because RAS is a protein that should only turn on time to time when the cell wants to decide, and right after that it should turn off by removing the last phosphate, and it should become inactive. If it is active all the time, it will induce cell division again, and here you have a tumor. So to prevent that, the RAS protein should be kept inactive by default. And of course, overactivation is very difficult, dangerous for cancer, but underactivation of the RAS protein is also dangerous, because it means the cell might die or might not grow, or the healing capacity of the tissue can decrease. So there should be a balance between the active and inactive RAS, which is under control of the receptor tyrosine kinase pathway. Yes. The next. Okay. This was one of the target, the RAS proteins. Another target of the receptor tyrosine kinase is the inositol phospholipid. Now, this is very similar to the previous case, the GPCR phosphatidylinositol here. So again, it's a membrane located signal. It is slightly different, but you don't have to memorize the difference. It is phosphorylated by default. And once the receptor tyrosine kinase, or maybe I should move this. Once the receptor tyrosine kinase is active, the PI3 kinase will bind to the scaffold protein with its SH2 domain. So it's going to activate the PI3 kinase. The PI3 kinase will activate the inositol phospholipid. Okay, as the name implies, it's a kinase. It is going to phosphorylate the target. Here the target has 2 phosphate group. It will add the 3rd phosphate groups, which means it's going to be active. Now, once it is 3-phosphorylated, it will also behave like a dog, and it is going to allow some proteins to bind and activate. So this is not an enzyme itself. It has no enzymatic activity, but it behaves like a dog, and it's going to allow interaction with some protein kinases. So these protein kinases, in return, activate AKT. Now, if you're going to study cancer, there are few words, there are few enzymes that you should always remember. One of them is AKT. This is a main signal. By the way, it's also called protein kinase B. Remember, there was the protein kinase A, which is activated by cyclic AMP. There was the protein kinase C, which is activated by the DAG and IP3. This is protein kinase B. And there is the MAP kinase, which is also activated under receptor tyrosine kinase. And there is RAS. So these are most central, most basic signals for cell division. If a cell is going to divide, it goes through this pathway. So in the case of cancers, we see a combination of these problems. So one of these enzymes are not working properly. Now, AKT is not only, we know that there is no such thing that this signal means that and this signal means that. There are combinations of signals. And in this context, we know that AKT is also used for survival. And I said that every cell has a gun. I'm not sure if I will be able to draw a gun pointed to itself. The trigger is ready to pull. Every day somebody should come and say, don't die today. If you don't say, don't die today, it's going to suicide. So the scientific name of the signal, don't die today, is survival signal. Again, it's a measure against cancer. So you have to tell every cell that they should survive that day. The day that you don't say that, it's going to suicide, which is also called apoptosis. By the way, I'm going to call, I might misuse these terms, apoptosis, etc. Ebru is going to tell them to you in much more details. Please do not be stubborn about the terminology. I'm just giving a very brief introduction. She's going to give them much more detail in the cell culture course. Okay. So what AKT does is that it interacts with the bud and BCL2. So because it's a kinase, it is going to phosphorylate this bud, and when it is phosphorylated, it becomes inactivated. Normally, it induces apoptosis. This way, it will not be able to induce apoptosis because it is inactive. And BCL2, it is going to promote survival. So it is the signal, don't die today. Okay? And it is the signal saying that don't suicide. AKT itself is important for survival, but when it is combined with other signal, it also means the divide now. Sorry. Or it can also mean grow now, because you know that growth, yes, is a prerequisite for division, but these two concepts are different. This is proliferation, which means cell will go under mitosis and make 2 copies of itself. This is simply getting larger. So although they are similar, growth and division are different things. So AKT combined with other things can mean any of them. Or it can also mean differentiation. Now, a third thing that you should always keep in mind is TOR. Now, TOR is another molecule that stimulates the growth in this case. We have talked about these local mediators, remember? The local mediators can be NGF, which is nerve growth factor, platelet growth factor, PDGF. These kind of local mediators, they induce growth in the target tissue. And how do they do that? They do it through receptor tyrosine kinase. And at the end, they activate TOR. And once the TOR is active, the protein synthesis is stimulated and protein degradation, which is done by the proteasome complex, is suppressed so that it is going to get bigger and bigger. And we know that TOR is also important for cancer. So some anti-cancer drugs, they block TOR to stop cancer, to stop growth. And this is an oversimplified, extremely simplified version of pathways. Please, please, please memorize these pathways. If you're ever going to study cancer, you're going to see all these pathways in details. And as you also see, sometimes you don't see it, but sometimes the pathways also talk between each other, because yes, they're induced by separate events, but they have to talk and communicate, sometimes make a common decision. Okay, I'm not going to go into the other one. So thank you for listening. Chapter 16 is